and it's my pleasure to introduce today you, Dr. Eva Pereiro. Eva Pereiro did her PhD at the European Synchrotron Radiation Facility in Grenoble, in France. After that, she joined ALBA Synchrotron, and at the end of 2005, to continue learning about the soft part of X-ray microscopy. In the process, she has visited several facilities that were leading this technique in Germany and United States. And back in Barcelona, Eva led the design and construction and operation of the Mistral beamline. And Eva's fun fact is that she continues working in Mistral beamline as she did forever and ever, trying to solve problems which pop up one day after the other with the user support, making her feel a bit like a firefighter. So keep fighting, Eva, please. Go for a while. And please go ahead with your talk.
So many thanks for the nice introduction. I'm just going to go to slide mode, sorry. So today I'm going to talk about X-rays, and I had a nice talk just before mine from Damian that has already introduced what is the technique. So my work is mainly done here. So I'm working at ALBA, which is a Synchrotron Spanish facility, and concretely in the Mistral beamline, which is dedicated to soft X-ray microscopy. And what we are trying to do is to look at the structure of the cells by tomography, and this is what I'm going to present here. So in my first slide, what you can see are different biological techniques, and I just wanted to put cryo-soft X-ray tomography in this frame because most of you might not know it. So cryo-soft X-ray tomography sits close to the 30 nanometer half-pitch resolution, and the thickness of the samples that we can image are about 10 micron. So as you all know, imaging across the resolution scales is necessary to understand complex biological systems, and this is what we have been seeing in the workshop in the last days, and I'm going to try to convince you that cryo-soft X-ray tomography is also a useful technique to do correlative approaches. So first of all, what is a transmission soft X-ray microscope? Well, it works like any other microscope. It has a condenser lens and an objective lens. The difference is that we are sitting far away from the source, about 26 meters from the source, and there are a lot of different instrumentation which is bringing the photons with the correct energy and with the correct dimensions, I would say, and coherence and divergence to feed the microscope. The achievable resolution is limited by the objective lens, which is a diffractive lens, a Fresnel zone plate lens concretely. And the data that we are acquiring are quantitative. And this is because the transmission signal is following what we call the Beer-Lambert law. And what that means is that the contrast that we are seeing in our images depends directly on the atoms that are on the sample on each voxel of our tomograms. So there is a trick that we are using, which is called the water window. This trick allows us to fit full cells without sectioning and without staining. And this is because we just image below the oxygen absorption edge. And for us, the water of the cell is transparent. So we do not need, again, this sectioning that is required, for instance, in electron microscopy and in electron tomography. So this is a typical, on the bottom right corner, this is a typical reconstruction slice where you can see different organelles of the cell. And these gray values that you see here are corresponding to the linear absorption coefficients of the atoms that you have in your voxels. So the workflow that we are following, although Damian has already shown you what we do at the beamline, we are using epifluorescence microscopy with dyes. This allows us to find the relevant cell or the relevant regions of the cell within the electron microscopic grids. So we are growing cells on electron microscopic grids, finder grids, or depositing them, depending on the type of cell you're looking at. We can do then what we call mosaics, so 2D projections of a full square where you will have, in general, several cells. And we are overlaying this with the fluorescence, visible light fluorescence. And this is a nice picture of one cell. You see very well the nucleus and then the cytoplasm more on the right side of the nucleus with the different dyes. This is what is allowing us to locate specific structures or molecules that we are interested in before starting doing the tomogram. Take into account that our field of view in the microscope to do a tomogram is more or less between 10 to 15 microns square. So this means that a full cell will not fit in your field of view, and you would need to use, for instance, stitching tomography to be able to have the full volume of the cell. So these are typical tomograms and typical information that we are getting. Again, on the upper corner, we have the 2D mosaic with the region in the yellow square that was imaged, which is on the right side, on the upper right side. You can see mitochondria, ER, you can see the plasma membrane, lipid droplets. Then on the bottom corner, for instance, you have the nucleus with the two nuclear membranes and nuclear pores. You have the nucleoli, nuclear membrane channels going through the nucleus. And on the right corner, you can see, for instance, that euchromatin and heterochromatin, because they have a different condensation, show a different gray value. So this is the typical images that we are getting: mitochondria, lipid droplets, endolysosomes, lysosomes, or autophagosomes. So last thing, this is a video showing one of the tomograms of a control cell. This is a human hepatocyte. You can see the ER going around the mitochondria, vesicles, the nucleus, sorry, is at the bottom left of this movie. And the last thing is, this is the typical resolution that we are getting in our volumes. So for this volume concretely, which was published recently, the resolution, the evaluation of the resolution achieved is 28 nanometer half pitch for the full volume. So this is very nice. Okay, what can we do with this? So I'm going to show you several examples. The first one would be showing pathogen infections, so to try to understand what happens during pathogen infections. Then I have chosen another example on biomineralization, and finally, two more examples to locate specific molecules within the cellular landscape that we can reveal with soft X-ray tomography. So the man of the day is Javier Conesa, because he was postdoc at the beamline. So I'm going to show several of his collaborative works. And this is the first one, together with people from the Instituto de Salud Carlos III and from the CNB. they were interested in understanding the asexual life cycle of a parasite called Babesia divergens. Babesia divergens is an apicomplex parasite that is causing babesiosis, which is transmitted by ticks to domestic animals, wild animals, and humans. And this parasite, which infects vertebrates, reproduce asexually by a multiplication of binary fission and budding within the erythrocytes, so red blood cells. And together with combining, confocal microscopy to get the chronological infection, what they were interested is trying to get all the main stages of the infection as well as the transient stages of the parasite. So the workflow is again the same. On the upper left image, you have the fluorescence, visible light fluorescence, to be able to choose which are the cells of interest. In this case, the red dye was a mitotracker. Then on the right upper picture, you have a 2D projection of the square, the same square on the left, which is in yellow. And then on the bottom left, you have the virtual slice of these red blood cells with the parasites inside, and the segmentation on the bottom right. So these are the main 7 intraerythrocytic stages. And I mean, I'm not an expert here, so I'm just going to mention them. You have the merozoite, which is the parasite outside the cell before infecting the red blood cell. Then comes the trophozoite, which is already inside the red blood cell. Then the double trophozoite, pair piriform, double pair piriform, the tetraeds, and the quadruple trophozoites. So you have the segmentation where you can follow the morphological changes in all these stages, where you have the nucleus in dark blue, the mitochondrion in light blue, the apicomplex in purple, dense granules in red, and the parasite membrane in yellow. And then in the inset, you have the GIMSA staining and the confocal picture of the same stage, of the same parasitic stage. And then in addition to the 7 main intraerythrocytic stages, you have many transient ones, and this is the full 4D, because it takes into account time, 4D reconstruction model of this asexual life cycle, which is quite complex, as you can see. So with this first example, what I wanted to show you is that we can follow cellular alterations due to parasitic infections at spatial resolutions that are around 25-30 nanometer half pitch. And of course, as we already know, understanding pathogen cycles in host cell is very important to design strategies to prevent diseases. And taking into account that we are in the middle of a pandemic, this is even more true with the real example life that we are all suffering from it. So my second example is regarding biomineralization. This is a collaboration with the Weizmann Institute of Science in Israel, where Keren Kahil was interested in the calcium transport of, in the calcium transport within sea urchin larvae. So the primary mesenchymian cells, which are the spicular building cells in these urchin larvae, endocytose seawater and integrate the calcium ions in a different network of vesicles, and little by little, the calcium is being concentrated until it precipitates in the form of amorphous calcium carbonate. And in the upper row of my picture here, what I'm showing is that if you tune the energy of the X-rays to the correct absorption edge, in this case for calcium is around 350 electron volts, then what you can get is a differential absorption due to the calcium atoms. And like this, if you do the difference of what is pre-edge, so just before the calcium absorption edge and at the calcium absorption edge, you get this type of pictures where you only see the calcium absorption within the cell. So this is very useful if you want to understand chemically what's going on, and this is what they did. You have several references first, and you do a spectra of this reference where you have seawater, hydrated amorphous calcium carbonate, anhydrous amorphous calcium carbonate, and calcite. Because the cells are very sensitive, you cannot really do full spectra on a cell, but what you use is a trick, which is to look at the intensity ratio between the pre-edge and the edge, because this intensity ratio is reflecting the calcium coordination shell, so that you can distinguish between the different chemical species. So this was done with these cells, with these primary mesenchymal cells, and what was shown is that there are many disordered phases, and most of them, these phases, were not known before. So this is an example with 2 cells. So 5 cells were analyzed. A total of 700 calcium-containing vesicles were analyzed. And what you can see in the cells, where the color code here of these vesicles correspond to this ratio with increasing calcium order, is that you have a continuum between the calcium ion of the seawater until the calcite. So there are very few calcite particles, and there are half of the calcium containing vesicles are between the level of the seawater and the hydrated amorphous calcium carbonate, while the other half of them are between the hydrated and the anhydrous amorphous calcium carbonate. So what I wanted to show you with this example is that we do not only have the 3D volume of the cell, where we can see the structures, but also chemical differentiation, which allow us with spectroscopy to get even more information on the cells. Another example, which was again led by Javier Conesa when he was postdoc at the Beamline, was this one that I'm showing here. This was a collaboration with the people from IMDEA Nanociencia in Madrid and the ESRF, the European Synchrotron Radiation Facility in France. So in this case, the goal here was to locate a specific iridium drug complex, which was very promising because it showed that it was more potent than cisplatin, okay, 10 to 200 times more potent. The action mechanism behind this drug is intracellular redox imbalance, so the creation of reactive oxygen species. And the design of the drug was such that the metal center was sheltered to avoid off-target reactivity, so that the drug could target to go to the final target position in the cell, let's say, before doing the being toxic. So for this research, what was applied was two different techniques. Again, this is soft X-ray tomography at Mistral beamline, and then with the same samples, we travel to Grenoble in France, and we measure hard X-ray fluorescence tomography. So the hard X-ray fluorescence has a very, very high sensitivity. It can detect ppm. And what we were doing was imaging the iridium signal on the same cells that were imaged at the Mistral beamline at ALBA. So here I'm showing one of the figures of this paper by Javier. On the left, you have the control cells, where you have the segmentation with, again, the nucleus in blue, dense vesicles in red, lipid droplets in green, vacuoles, mitochondria in red, and the cell membrane. And on the right side, what we could see was that after 12 hours and with 1 micromolar of this complex, which is called ACC25, we could already see some hallmarks of cell death, which were, as you can see in the mitochondria segmentation, swelling and fragmentation of mitochondria. If you go to classical EM epoxy resin imaging, you will see that in addition to this mitochondria swelling and fragmentation, you also have Golgi vacuolization and ER disruption, which again indicates that the cell death. So now after imaging, at Mistral and having the tomograms of the interesting cells, such as the one that I'm showing here, we did hard X-ray fluorescence tomography on the same cell, mapping exclusively the iron signal. In the upper row here, what you can see is on the left upper corner, you have again a 2D mosaic of the view of the cell overlay with the visible light fluorescence signal coming from mitotracker and lysotracker. On the middle, you have the mapping, the 2D mapping of the iridium signal. And what we can see on the third image on the upper right corner is that this iridium signal seems to fit with the mitotracker signal that we get on the epifluorescence microscope. But of course, this is 2D, which means it's a projection. So you really cannot say that this is the reality. So we need to go to 3D and we need to see where the iridium signal is coming from. And from that, when you overlay the mapping, the 3D mapping of the iridium signal with the soft X-ray tomography, what you can see is that the iridium signal is exclusively content in the mitochondria. And this is what is shown in the segmentation on the right part of the figure, where you can see again the mesh, the external surface of the mitochondria, which is a yellow mesh, with the quantification of the signal of the iridium, which is found exclusively within these mitochondria. And Javier did a very nice movie of this result and this combination of techniques. So we were using silicon nitride membranes because grids are not very good for hard X-ray fluorescence. And you can see that you were seeing before the mitotracker signal. This is the tomogram of the full cell. This is a stitching really of 7 tomograms to get the full volume of the cell. And now you see the segmentation with the nucleus, the dense vesicles in red, lipid droplets in green, vacuoles in white, and the mitochondria in yellow. And what you will see now is the same volume with the signal of the iridium in purple. Well, not all is purple, it will depend on the quantity of iridium found. And this iridium is exclusively found in the mitochondria. So this is a very important achievement because drug localization is necessary to understand the cytotoxic mechanism and also to improve design strategies to have better drugs for better therapies. So my last example, and I'm hoping I'm not too late, is a work in progress, and this is done by Jan Groen, who is PhD student at the Beamline. We are again interested here in localizing a specific molecule, which is a protein repeat structure that we use to inhibit collagen formation in fibroblasts, but I'm not going to show you really the research, but the combination of techniques here. So what we did was to combine 3D SIM in cryogenic conditions with soft X-ray tomography on the same cell. There was quite a lot of work in fiducialization because you will finish with 2 different volumes which have different dimensions, different resolutions, and you need to be very accurate when you're trying to get the 2 volumes together and have the overlay in 3D. So I'm showing you here just one example of fiducials that we were using, tetraspecs, which are they show a 200 nanometer fluorescent signal with a core that is between 80 and 90 nanometers that we can see in soft X-ray tomography. So these tetraspecs can be seen in fluorescence and in absorption contrast, which is very useful for us. And this this is shown, at the bottom right corner of my picture, where you can see the dark core, which we are imaging in tomography, and the green signal coming from the fluorescence. Of course, the 3D SIM that we are using in cryo has a lateral resolution of 200 nanometers, which is less than what we are having with soft X-ray tomography, and an actual resolution that is worse than the lateral one of about 500 nanometers. In this case, we were collaborating with Diamond because they have been developing this cryo 3D SIM, and we are now following their strategy and developing this also at ALBA. So this is to show you another volume where you can see on the left the reconstructed tomogram with the mitochondria and the nucleus, and in the bottom of my picture. The middle figure is the 3D SIM mitotracker signal and the overlay of both of them. It is very useful when you try to correlate to have not only fiducials outside the cell, but also, let's say, organelles that can be used as fiducial markers as well, because in your volume, you will have the fiducials on top of the cell, but then you cannot really correlate anything within the cell. So this is why MitoTracker, for instance, can be a very nice way to correlate structures. The SIM, in addition to that, is a very nice instrument for us because we are able to screen several grids. It's a fast technique. So we can screen several grids in one day and then go to the transmission X-ray microscope and image those grids just after that. So this is a final image that I'm going to show. This is the full on the right. You have the overlay of the SIM signal volume. For the full volume, I'm only showing one of the planes, and the tomograms that we took for this cell, which are 5 of them. And on the left part, you can see on the bottom only the MitoTracker signal, and on the green was the signal by our protein. This is what we wanted to localize, and we have been localizing it on specific vesicles, which we don't know yet what they are. So I think there is a movie, yeah. I'm not sure it's starting now. So this is going through the volume with the two combined data sets, the fluorescence and the soft X-ray tomogram. So as a conclusion of this part, it is very, very important for us to work in correlative approaches with visible light microscopy, mainly because obviously most of the biolabs worldwide are using visible light microscopy as workhorse tools to localize their specific proteins and structures and so on. So the way for us to go at ALBA now is to develop this Cryo 3D SIM to be able to offer it to our users just prior to the beam time, the X-ray beam time. It is necessary to do this in cryo because the workflow, if you're going from room temperature to cryo, you will probably will not find anymore the same cells after vitrification. Things are moving and so on. So all the workflow should be done at cryo temperatures. And of course, it would be nice that in the next years, there is push, let's say, for super resolution techniques, which would be in cryo, but with resolutions better than 100 nanometers. So close to my final slide, sorry. I hope I have convinced you that soft X-ray tomography is a complementary technique to electron and visible light microscopies, but also to hard X-ray fluorescence, which can be extremely useful. Cryocorrelative approaches have been shown in all the workshop, and especially with the talk of Paul this morning. This is the way to go to try to answer complex cellular questions. Developing 3D cryo-super resolution techniques is something mandatory, and we're working on that. And finally, I will mention all the people that have been involved in this work. Javier Conesa, who was at the Beamline until May 2019, now is at CNB. Anna, Andrea, and Jan. We are only 4 at the Beamline, so the progress is low, unfortunately. Our Weizmann collaborators, Erin, Leah, and Steve, collaborators at the Instituto de Salud Carlos III, Daniel Luque, Estrella Montero, Pepe Carrascosa, of course, at the CNB, the 2 IMDEA ladies, Ana Carrasco and Ana Pizarro, the people from the SRF, Peter Klutens and Yang Yang, and finally, Maria Archeolati from Diamond for letting us use the SIM. And I'll thank you for your attention. So I'm
okay, Eva, thanks for the talk. Yeah, we have some pasted in the chat in the Hopin platform. We have two questions from Paul. The first one is, what could be the constraints for using tissue? And the thickness, the tube shape?
So we cannot measure more than 10 microns. So if you want to use tissue, you would need to cut the tissue. So that's something that is a fundamental limit. More than 10 microns, you are not getting enough transmission to get a proper tomogram anymore. So of course you would need high pressure freezing and then sectioning of the tissue and keeping everything in cryo conditions.
Okay, we have an additional question from Paul, is that if you can discriminate between vitreous water and ice crystals?
Well, we will see ice crystals at our resolution. Of course, if there is small little ice crystals which are less than 30 nanometers, we will not be able to distinguish them. The point is that, as you know, when you have a change of phase between vitreous ice to ice crystals, what happens is that the membranes and so on will get disrupted because the volume of the cubic crystal will increase. But of course, I mean, our resolution is what it is, 30 nanometers. And if there's something smaller than that, we will not distinguish it. We do see crystals formation mainly on top of the samples when you have contamination, when you're moving the grid around and so on. And this is clearly visible, and you can see the little cubic structure. But within the sample at the moment, when you have a devitrification, what we can see in general is that the image becomes fuzzy, that the membranes are becoming completely fuzzy. And this indicates that there is some devitrification. But we haven't seen yet formation of ice at our resolution inside the cell.
Thanks, Eva. And the last two questions are from Erin, and they are both related with the tomogram stitching. So, how long does it take to acquire and process 7 tomograms? And if it's possible to montage these tomograms during the acquisition of the T series, as is possible in electron tomography?
Okay, so the tomogram, I haven't mentioned that, but it's few minutes collecting the tomogram. So this is pretty fast. Then we have an automatic alignment of the tomogram at the beamline, but the automatic alignment depends on the gold fiducials that you're using to align projections together. So if the spread of these gold fiducials is not good, your automatic alignment will not work, and then you will need to rework your alignment after the beam time. If it's good, then you will have the tomogram ready, the full volume reconstructed in about, I don't know, 8 minutes, let's say. And then for the stitching, this is much more complicated. We are using IC-clamp for the stitching, and then you need a lot of manual work, and this is in general not done at the beamline, but already when the users are back at their facilities, at their laboratories, because it takes quite a lot of more time than simple reconstruction.
Okay, so we have no more time for questions, so we can keep going in the meet the speakers after the next talk. And thanks again, Eva.
Thank
for
you
the
for
nice talk.
inviting me.